So there are really two functions of the genetic material within a cell. In the later videos, I'll talk about how we use the genetic information to code for proteins. But in this video, I'm going to talk about replication. So when a cell is under favorable conditions, a prokaryotic cell has plenty of nutrients around, it is going to reproduce or make copies of itself. So remember that process in bacteria is called binary fission, where essentially you have a single cell that will copy its genetic information and then it will cleave in half, and you will have two daughter cells that are genetically identical to one another and are genetically identical pretty much to the parent cell. So cells have to be able to replicate or make perfect copies of their DNA. And this takes a lot of different enzymes, and we're only going to narrow it down to a few of the most important. But the general idea is that we need to first separate the strands, and then we need to copy the template, and then we produce and separate out into two daughter molecules. So the five enzymes that are responsible for that process are helicase, primase, DNA polymerase 3, DNA polymerase 1, and ligase. And I'll talk more in detail as I kind of go through the process of replication. But helicase is when its job is to unzip the DNA, so to open it up and expose it. Primase synthesizes an RNA primer, and I'll talk about why this is necessary in a minute. DNA polymerase 3 is responsible for creating and building the new strand by reading what is the template, right, and attaching the correct bases in order to make a copy. And sometimes it makes mistakes, so it also can help to proofread its mistakes. Now, DNA polymerase 1, its job is to remove the primer, and we'll talk about why that is later, close gaps, and we'll talk about how those arise, and repair any mismatches that may have arisen. I do a little bit of proofreading. And then ligase finalizes by binding together the pieces of DNA at the end. All right, so again, this is just a real quick overview. Now I'm going to go through this whole process in detail, utilizing and describing where these enzymes are in play. All right, so really what has to happen is we have the parent DNA, double-stranded piece of DNA, and we want to make two exact copies, right? So the parent DNA molecule must first sort of uncoil itself and then open up so we can read the strands. And there are hydrogen bonds, remember, in between the nitrogenous bases, and those are those weak bonds. And so we can unzip or open up and separate the two strands. Helicase is the enzyme whose job it is. Then as we sort of bust open the DNA and separate out or unzip, then we can read either side of the DNA and create a new strand, single strand on either side, to build a new piece of DNA. And we'll look at it in images here in a second. Maybe then it'll make a little bit more sense. All right, so where does this typically start? Where do we start to unzip the DNA? Well, the helicase is going to come in and they're going to find a segment in the DNA where there's a lot of A's and T's, right? So if you look here, this is adenine, this is thymine. Adenine and thymine only have two hydrogen bonds, where cytosine and guanine have three. So if you have three bonds, you're a little bit more difficult to pull apart. It's just a little bit stronger because you've got more of them. So helicase finds an area where there's lots of A's and T's, and it untwists and breaks those bonds, those hydrogen bonds between the A's and T's, and basically opens up the two strands so we can start the process of replication. So you can see here that here you've got your double-stranded piece of DNA. Helicase comes in and pushes open the sides, right? So we can read the template strand. Next is going to come along a primer and DNA polymerase, which then is going to go along either side of the strand and actually start to make the copy. So the pink strand is the new stuff, and the blue strand is the old stuff. Okay. So DNA polymerase 3 is the enzyme that builds the new strand. However, DNA polymerase 3 has a little bit of a problem with it. There's actually like two different problems. One, it can't just start building a new strand with nothing. Like it can't just start putting together nucleotides. It has to attach a nucleotide to something. The other problem is that it can only synthesize in one direction, right? So the reason why we have this little piece right here is the RNA primer. So primase is an enzyme that makes a tiny little piece of RNA that has a few base pairs, right? So it might be an A, a U, and a G or something, right? Just a few base pairs that is close enough that it sort of clings on to this like a magnet. It'll just recognize a part of the DNA and it'll adhere onto that. Then the DNA polymerase 3, which is this yellow blob, can come and start building the strand, right? Now, that solves the problem that we have with DNA polymerase 3 that it needs something to attach to. So we have this little RNA primer that's sitting and waiting for DNA polymerase 3 to start attaching and building the new sequence. The other thing is that it can only go in one direction. DNA polymerase 3 can only bind in the 5 to 3' direction. So looking at this image, you see that, one DNA polymerase 3 is going this way, the other is going this way. And we'll actually look and see how it looks a little bit different. Bacteria are a little bit better at doing this, but just remember that DNA polymerase 3 can only build in one direction from 5' to 3', right? All right, so this is a little bit more reflective of how the DNA polymerase 3 looks. On the other image, we saw one on each side, but really what happens is the DNA polymerase 3 will come in and it'll wedge the circular chromosome open, and it crosses over both sides of the strand. So as it starts to code and go around the outside of the chromosome, it's actually working on both sides of the strand at one time. So this right here is called the replication fork. And there's a whole video of me explaining this in detail and walking through all of this as well. So you can go and watch that if you want a little bit more detail about that. But what ends up happening is the DNA polymerase works its way down and around. And because we have the anti-parallel arrangement, that means that the DNA polymerase must be coding and making a new strand on one side that's in the correct direction, the 5' to 3' that it can build, but on the other end it must be going backwards. So we know that it can't actually build backwards. So what ends up happening is the top of the enzyme creates this nice smooth strand that is in the right direction, and it's 5' to 3', and we call it the leading strand. Whereas the other end has to sort of hop forward and code backwards, and then hop forward and code backwards, and hop forward and code backwards. And as it does that, it makes little snips or little pieces of DNA, and this is called the lagging strand, and those little fragments that it makes are called Okazaki fragments. And it will have to come back, we'll have to come back after the whole replication is done to sort of stick those together, right? So on each side, there's going to be a lagging and a leading strand because the DNA polymerase is going to be going around to the bottom, right? Now, DNA polymerase 3 can proofread if it makes any mistakes, but DNA polymerase 1 also can do that. It can also fill in if like DNA polymerase 1 or 3 forgets to put in a nucleotide somewhere, it can stick some in there. It also will take off the RNA primer and replace it with DNA, right? So remember, that's the little RNA primer right here. So DNA polymerase 1 is going to have to come along and like get rid of that and put nucleotides there. So DNA polymerase 1 really is in charge of doing little, not building big long strands, but making little corrections and filling in little holes. And then the last thing that happens is we have to be able to seal together the Okazaki fragments. And so ligase is what does that. So that's the enzyme that does the last sealing of the lagging strand and sort of finalization and separation of the two daughter molecules. So you see what happens is we end up with two identical chromosomes, right? One of the sides of the chromosome comes from the original parent cell, and then the other comes from the new cell. So, or it's the new strand, right? So as this sort of divides and separates, you can see how there would be one circular chromosome that's got a blue side and a pink side, and then another one that has a blue side and a pink side, the pink being the new and the blue being the old. And so it separates out, and each daughter cell will have a side that comes from the original parent, and then a side that was newly built, and then the other daughter molecule would have a side from the original parent, and then a new that was newly built. Right? Now, there may be some mistakes that happen along the way where we've either added the wrong base or skipped or added an extra one in. So there's lots of different mutations. Most of these mutations are going to be corrected by DNA polymerase 3 or DNA polymerase 1, but if they're not corrected, it results in what's called a point mutation, and we'll talk about how that can lead to problems when we or benefits or do really nothing. So what do those point mutations mean in terms of the phenotype and the translation into proteins? So again, just to kind of take a look at, these are the 5 enzymes that I'm going to expect you to know. So helicase is the unzipping, primase makes the little RNA primer so that DNA polymerase 3 can start to build the strand, proofreading itself as it goes along. DNA polymerase 1 will remove that primer and replace it with DNA, close any gaps or repair any problems that DNA polymerase 3 may have left behind, and then ligase finalizes and separates out the chromosomes so that the whole cell can cleave and complete the replication of the total cell by the process of binary fission.